Alright, so I just went to the -80 freezer and got these competent cells that we buy from a company, and they need to thaw on ice for 6 minutes. So they're on ice now, I don't need to do anything, we'll wait for 6 minutes and then come back. Alright, it's been 6 minutes, the cells should be thawed now, so what we need to do is take the cells, the competent cells, and add them to the DNA. So I have a pipetter set for 50 microliters, 50, and I'm going to take this tube here. The hardest part of this is getting these lids off. They can be really tough, but that one came off easily. So I'm going to take the cells, all 50 microliters, dispose of that, and now I'm going to add it to my Golden Gate assembly that came out of the thermocycler, and I'm just going to put it in there very gently and then quickly put it back on ice. So that was my experimental. I need to do the same thing with the negative control, and then we also have a positive control. And this positive control did not have to go through Golden Gate assembly. It's already a built plasmid that has a promoter, RBS, and RFP, and we know it will produce red fluorescent protein. So I'll transform those two into cells and then come back after 5 minutes of letting all of that sit on ice. Alright, it's been 5 more minutes now, so I'm going to adjust this, and we're going to add SOC media, this really rich media, to the cells. So I'm going to get a fresh tip, take out my 30 microliters. I prefer to use my prehensile pinky. Got 30 microliters, always protect the stock, close that up. And now I'm going to add it to the cell and DNA mixture. And that's good to go. So I will do that to the other 2 tubes and all the tubes for all the transformations. And my mask is slipping. All right, and then after that, we're going to plate them onto plates. Sorry.